Alright, so now that you guys know of how to make, theoretically, transgenic mice and also knockout mice, let's basically look at the differences or compare them, transgenic versus knockout. So in transgenic model, most of the time it is for gain of function mutations, and sometimes you can also do loss of function, but loss of function most of the time is for knockout. Because in knockout we know that the function is to knock off the gene or turn off the gene function. Secondly, in transgenic model, genes are added into the genome to express certain particular protein or proteins, but for knockout model, genes are inactivated by deletion or expression of mutant copy. I mean, of course, the mutant copy doesn't really have the expression of the original gene. And the third differences or comparison between them is that for transgenic model, uh, most of the time integration of the transgene is by random integration. But for knockout, we know that from the slides early on, you basically need to have a targeted integration of the gene into a site-specific genome. So those are the comparison between transgenic versus knockout. So let's move on to the three knocks. So way earlier, I've basically told you guys that there are three knocks apart from the knockout that you may have heard early on. So well, and also we have covered on knockout. So knockout is basically when you replace or disrupt the actual function of the gene, and this is usually basically for drug selection. And most of the time, mice could be used for knockout study, but usually to study only the effect of the loss of a gene, not a specific mutation. So let's have a look at what is knock-in. So as you can see over here, knock-in is where a mutated DNA sequence is exchanged for the endogenous sequence without any further disruption of the gene. So what it means is that you basically swap function or swap some of the coding region of the gene. So some knock-in strategies basically rely on the use of gene vectors with flanking sequences such as LOXP, and of course, based on exposure to a certain enzyme such as Cre recombinase. So this kind of recombination sometimes result in deletion of the intervening DNA, which could be functional, which could not be functional, but it depends on the result of the study. So using the knock-in method, it is actually possible to replace a gene sequence with a sequence of researcher's choice, and of course to delete unnecessary sequences, whether it has a certain function or whether it's just there for reason of being there. And the last one among the three knocks, knockdown. So knockdown is achieved by using RNAi or RNA interference. Hopefully some of you have heard of RNAi. So what it means that by using RNAi is that genes are silenced or knocked down by short pieces of double-stranded RNA. So we know that RNA is single-stranded, but in this case we are using double-stranded RNA. So basically, RNAi opened up a lot of exploration, and the discovery has expanded to the concept of heritable regulators of gene expression, to include, of course, RNA molecule. Now, using this method, most of the time viral vectors is being used because we know that some viruses has RNA as a genome. So, insertion of interfering RNA into the stem cells is done using a virus as a vehicle to deliver for reconstitution. Or sometimes, if you are not using viral vectors, you can basically just modify the activity of the genes in selected tissues. So, in this diagram, this is actually on the differences between knockout and knock-in. So, if you were to look up on knockdown over the internet or in Google, probably not much information is on knockdown, because knockdown is actually more or less better known as RNA interference study. So that's why if you were to compare all the three knocks, usually knockout and knock-in are being compared. And as you can see, gene sequence or the transient construction are different between them. Right? Now, these are the comparison of the three knocks, knockout, knockdown, and also knock-in. So basically by looking at this, and I hope I did put this in your notes, you can more see on the differences and try to find similarities or what is shared between the three of them, because your next activity is actually on these three knocks. Alright, so activity 5. Now before anyone asks me, we last stop at activity 3 on Tuesday, but there is no activity 4. So activity 4 is actually on a quiz, but we're seeing that we are doing it online, so I'm not going to do any quiz online. So we just move on to activity 5. Activity 5 is of course an individual activity, whereby I would like to know the similarities between knockout, knock-in, and knockdown, right? So write your answer on the web link in your e-learning. I will open our portal, most probably by using Padlet. So you guys are used to Padlet, so just put in your answer over there. Okay? So this activity basically is ongoing until, and probably we'll discuss it after your test. Alright, so now let's have a look at some other transgenic animal apart from transgenic mice. So if you remember in earlier videos, probably last week, I was saying that to produce transgenic animal, the larger the size, the harder it is. Okay, but actually it could still be done. So the first transgenic cow, okay, were born in 1998. This is basically achieved based on the success of some of the techniques. So we know that, okay, early in 1980s, it is done via microinjection. This is what they have tried, but without much success actually. So the only source of fertilized bovine zygote, okay, was basically from superovulated cows. The same keyword, superovulated, and the zygote again has to be recovered surgically or slaughtered. So the problem here is that they lose the cow, and they have to basically kind of like, well, it is a huge loss, okay, because to grow a cow or to have cow at a certain age is actually takes some time. So secondly, donor cow, okay, can be used only once and produce low usable embryos, so downside as well. Third, microinjection has to be done in over 1,000 embryos from more than 300 cows in about 150 recipients for a single calf production. And most of the offspring born were not transgenic, even after interbreeding, and the transgenic did not express the gene or pass it on to the next generation. So later on in 1980s, still by using microinjection, they developed oocyte in vitro maturation, and of course try to do the fertilization system in vitro as well. So in vitro culture system was used to make sure that embryo grows, okay, from the blastocyst to a reasonable production rate instead of just taking it from the cow at a later stage. So production of transgenic cow can be done, but at very low cost, and it basically saves some of the cows, okay, because they are not being slaughtered and whatever. But what they found that, okay, microinjection could result in detrimental mutation and variations of gene expression. And of course, it takes about more than 5 years, okay, to get a homozygous line from the founder. Usually more than that, about 7 years. So what they come up with, okay, to improve it is by using cloning. So they use somatic cell nuclei to clone cow to full term, and the DNA is replaced in an unfertilized oocyte containing DNA from somatic cell, which of course later developed into an embryo. So cloning is only used to produce the identical calves, but genetic manipulation of somatic cell enables the production of transgenic cow. So when compared to microinjection, only about 10 to 15 recipients are needed, hence reducing the cost. So the method using here is actually by nuclear transplantation. So how do they do it? Researchers grew bovine fetal fibroblast cells in a lab, and antibiotic-resistant marker was added. Nuclei from survivors were fused with enucleated cow eggs, nucleus removed, and employ Dolly technique. So these are the techniques done. So first step is that cell lines were isolated and cultured in vitro. So they only use high quality cows and make sure that the embryos are recovered after insemination with artificially cow, very good bull and whatnot. So once everything is okay, transferred to surrogate mother for 90 days, and then the embryos are killed to isolate fibroblast cells, and the cell lines are produced, and its sex determined pre-culture. The next thing is on DNA isolation. So the first and the second step could be done simultaneously, or it depends on the researcher whether which one they want to do it first. So it's interchangeable. So DNA isolation is also done, whereby it is extracted from cell lines and the library is prepared. So they compare of existing cattle DNA with the prepared library, and suitable sequences are isolated and further cloned. So the next thing to be done is on gene modification from the DNA, whereby modified cultured cell lines DNA are done and integrated with antibiotic resistance marker. So when exposed to this antibiotic, the altered gene containing the marker will survive and of course separated from those without the altered gene. Step 4. Implantation of the genetically modified embryos. So transgenic cells injected into enucleated cattle eggs and of course are then fused using nuclear transfer. So the embryos are cultured in vitro and then transferred to the surrogate mother implanted. So surrogate cows are monitored, and those who fail to be pregnant after 3 trials are removed. Remove meaning from the program or from the experiment. So the calves from pregnant cows are delivered via cesarean section. They actually do not allow it to be a normal delivery. Now the 5th step is induction of lactation, whereby transgenic calves are induced into lactation at around 6 to 9 months. Level of transgenic protein in the milk are determined. Embryos from the eggs are identified for female carrier of the transgene, and the embryos will be transferred into surrogate mother. So as you can see, the steps are a bit more complex as compared when you are producing transgenic mice, because this is a larger animal, so they want to make sure that nothing is wrong and they do not cause suffering to the animal. Alright, so recent advancement to cloning in cows is by gene targeting. So they use somatic cell cloning technology by inserting a transgene at a targeted site of host DNA. So it could be used to inactivate gene or insert new genes into predetermined site and replace gene variation, meaning that the 3 knocks could be done as well. Male and female founder animals could be made, and a homozygous transgenic calf can be made without germline transmission by inserting the transgene into both pairs of the chromosome. And now that we have the CRISPR technique, this is made even possible. Second is by microchromosome transfer. So chromosome, I'm sure you guys know chromosome is. Microchromosomes are basically a part of the actual chromosome. So they are small number of chromosomes or small size chromosomes. So they actually consist of millions of DNA bases containing hundreds of genes. Microchromosomes could replicate on its own, and of course are carried along during cell division as independent chromosome. So the insertion of human-derived microchromosome is made possible. Now, apart from that, a company in the US called Gala Biotech basically has patented a process called transgenic. So as you can see over here, there is an R there, means that it has been registered, it has been trademarked. So you can just Google Gala Biotech, it will bring you to the company website. So they are doing or basically producing transgenic cow using developed gene construct and vectors. Genes are inserted as stable single copies and inserted more efficiently. How, they do not disclose it because they have patented it. Scalability, which means that ERTs can be increased through conventional breeding, no problem there. And containment and control are facilitated during the interbreeding. So now the benefits of the transgenic cows are among them to produce more casein, produce non-allergenic protein for those who allergic to protein for whatever reason apart from lactose intolerant, produce a drug for treating multiple sclerosis, and we actually will see about this in farming, PAGRMING, provide techniques for producing other disease-fighting drugs, for incorporating medicines into food as well. It is also done to help scientists to understand how milk protein is produced and modified, and also to study the physical or physiochemical properties of bovine milk with different compositions. So let's have a look at some other transgenic animals. I think I have transgenic fish and transgenic chicken. So let's have a look at transgenic fish. So it is actually easier to develop transgenic fish, and it does not need high technology approaches. The eggs could be obtained easily just by squeezing the female, because, well, there are eggs again next to the gill, so basically you can just squeeze it out. Common method is by electroporation, whereby the egg cells again are electroporated with the desired DNA so that it is fused together and more or less go through mitosis. Other approach that could be used is to grow both male and female hormones in bacteria and utilize it. And usually after hatching, the male fish can become female in appearance because of the hormone, but of course maintain its male chromosome. So in the second approach, it creates a pseudomale, and the pseudomale can lay eggs with viable offspring, which means that the male becomes female now, even though it still has the male chromosome. So the female fish can also be sex reversed to male when exposed to hormone to become reproductively viable pseudomale, meaning that they have a win-win situation, that they produces more fish. So the transgene are usually directly injected or mixed with fertilized fish eggs by absorption. Just like any other transgenic animal, of course, the eggs will hatch to produce genetically changed and normal fish. So the first genetically engineered Atlantic salmon can reach around 7 to 10 pounds in 40 months or less, so it's roughly less than 5 kilos. These fish usually require more oxygen and food on a daily basis, but still less than the total food consumed by normal salmon. Then those are the observations that has been done. So now, these are some of the examples of transgene introduced into fish, basically just to see on the effect on the phenotypes. So I'm not going to dwell too much. This is in your notes. You can just go through it if you're interested. So, I think before chicken, I have transgenic sheep. So we know that Dolly is a sheep, so it has been created, and it is done by inserting transgene at random. So, in a recent development, a transgene was successfully inserted into a specific locus. So this is how it is done. The sheep fibroblasts were treated with a vector with a few encoding gene, a specific promoter site, and ribosome binding site. So only the successfully transformed cells are fused with enucleated sheep eggs and implanted into the surrogate ewe. And once the embryo survive, and of course several of them will survive, so they are being used, and of course, two young calves at least are being produced because then they want to mate it, for the interbreeding. So the two lambs were found to secrete milk with large amounts of product from transgene. So this is the first successful transgenic sheep since Dolly. So I think this is the last one, transgenic chicken. So you know that chicken also grows faster than sheep, probably not as fast as fish, but still faster than sheep or cow. So there are two methods to produce transgenic chicken. The first is to infect embryo with viral vector by basically carrying human gene for therapeutic protein, and also promoter sequence that will respond to signals for protein production. This is more or less for farming. Secondly, by transforming rooster sperm with a human gene and the appropriate promoters and checking for any transgenic offspring. So results from both methods have been tested and showed the possibility of producing about 0.1 gram of human protein indirect, because we consume the eggs, right? So we can get some benefit out of it. Alright, so I think I'll just stop it here and you guys can proceed with the next video.